This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. You're listening to TWiM, the weekly discussion show that explores unseen life on Earth. I'm Vincent Racaniello. This is episode number 31, and today is April 12th, 2012. Joining me today to talk all about microbes from Yale University, Joe Handelsman.
Hi, Vincent.
How are you, Joe? We haven't heard you in a while.
It's good to be back.
Did you miss us?
I missed you terribly.
Good.
We missed you.
Oh, that's good to hear.
Yeah, it's a few times, or last time, I guess. We were expecting you, and you couldn't make it. We were very disappointed. I'd like to have a little change in the cast here.
That's always
Makes
good.
It more interesting. So I'm glad you could make it today. I know you have a tough schedule. Also joining us today from the Medical University of South Carolina, Michael Schmidt.
Good afternoon, everyone. How's
How are
everyone doing?
Doing great. Really nice day today.
Yes, I think the whole country has been blessed by good weather.
Oh, except New Haven. We had a sudden spout of rain today, totally unexpected. So I guess we've got the one cloud in the country.
Well, you could use the rain. It's been pretty dry around here.
Yeah, there have actually been some brush fires, which I thought was pretty unusual for Connecticut.
Well, yesterday at the Meadowlands, there was a huge brush fire. I don't know if you know what that looks like. It's all brush, right, and flat. And the firemen couldn't get in to fight it because it was just burning kind of crazy. They actually closed the New Jersey Turnpike for a while.
Wow.
Big black cloud of smoke going into the sky. Pretty dry. Well, today, as usual, we have two really neat stories to tell you about. And we will start with a paper published in Nature Structural and Molecular Biology, a relatively new Nature journal. They just proliferate like crazy, these new journals. This one is called Discovery of an Archetypal Protein Transport System in Bacterial Outer Membranes. And I thought this would be interesting. We talked a little bit about transport systems with Stan a few weeks ago, and we talked about the phage spike protein. I don't know if either one of you saw that paper, the structure of a phage spike protein and how it punches through the membrane. It's really neat. And they have homologies to some of these bacterial transport, or these secretion systems, I should say. And also, I thought that Michael would like this. This seemed to be right up your alley, Michael.
And it takes me back to my postdoc years where I was working on bacterial protein export in E. coli a long time ago. And, you know, bacterial protein export is truly remarkable when you think about it. Bacteria, unlike eukaryotic cells that have all these wonderful compartments, bacteria are effectively a bag of enzymes. And, you know, the purists out there we're gonna get letters for me calling them a bag of enzymes.
Hey, Michael, we had an episode titled "Not Just a Bag of Enzymes." Remember?
I do. I do. And, you know, the issue is that protein synthesis occurs in the cytoplasm. And, you know, the molecular or the early days of molecular biology companies that were trying to express recombinant proteins, one of the early things they recognized is that when they hooked up their gene of choice to their super duper promoter and really cranked up the synthesis, they were very adept at making rocks. And these rocks were actually the recombinant proteins that they were trying to get. And so a lot of effort was really expended in trying to understand how to coordinate the prevention of rock formation, or these inclusion bodies inside the bacterial cell, for when you make proteins. Every protein has a particular octanol water coefficient that enables it to fold into the proper state. And inside a cell, there's a particular octanol water coefficient that, you know, each protein develops, and you can effectively end up with rocks or not. But bacteria have long struggled with sending proteins out, and we've had discussions about type 3 secretion and how that magic occurs in bacteria to coordinate and regulate virulence, and where the bacteria effectively interact and make molecular syringes and do all sorts of remarkable things, and that's effectively type 3 secretion. I did my early work on type 2 secretion, where principally it's the Sec system, or Sec A and all of its remarkable players through Sec Y, and that's the general bacterial protein export system, where it begins to move proteins into the membrane, the periplasmic space, as well as the outer membrane. And today we're going to be discussing this this group of transport systems that really are pretty remarkable in that they're often referred to as autotransporters. And
why is that? Do they bring cars across the membrane?
Yeah, pretty much. Well, as the microbiologists are really clever people in that we like to operationally define things. And an autotransporter is, or was, operationally defined as a self-contained secretion machine. And when you begin to look at the cool figures that they have in the paper, principally you have figure 1 that talks about the protein, that the system that we are going to be discussing today, which is TAM, which is the abbreviation for a translocation and assembly module. You'll begin to understand what we're going to be talking about, because these autotransporters were operationally defined as a self-contained secretion machine in Proteobacter or gram-negatives, where the N-terminal passenger domain would be translocated, which is code for move, from the cytoplasm across the first layer of grease, which is the outer membrane. Well, you're laughing, but
it's great. I love it.
Think about, go back to your basic biochemistry and remember the whole issue of hydrophilic amino acids, the ones that love water, hydrophobic amino acids, the ones that hate water, and then you have the amino acids in between, the ones that are charged, the positively charged ones, things like glutamine and aspartamine and all of these other positively charged amino acids. Then you have the kinky amino acid like proline that effectively bends things. And so what happens in these autotransporters is the N-terminal passenger domain is going to move or be translocated across the membrane through its own, and this is the cool part, where the C-terminal end of this protein forms this beta barrel domain that effectively, in the way I look at it, is it creates a door that the N-terminal passenger domain can actually move through these layers in which proteins or amino acids would effectively get stuck. And there were some really elegant studies done in the 90s by Colin Manol, where he actually had fusions, and he was able to make these wonderful fusions and actually begin to study membrane proteins by targeting and how they move proteins through the cytoplasmic membrane, and you could show topology of proteins. And so this is really building on the work of many investigators. And in this bacterial secretion machine that we're going to discuss here today is, or what these authors are describing, as a fundamentally discrete or distinct secretion process.
Now, well, you said that the part of the protein goes through this beta barrel, right?
Right.
And does it carry something with it to bring across the membrane, or is it just the protein itself going across that's the functional aspect?
Well, like all things in biology, it depends. Some of them just, move themselves.
Mm-hmm.
The good examples of some of these autotransporters are like homolysins.
I see. Right.
And,
Poke holes. Yeah.
yeah, they poke holes. And in this particular case that we're going to be talking about today, and this is where the authors are sort of the take-home message of why do this research. And the reason for it is this secretion machine that we're going to talk about will actually be an ideal candidate or an ideal target for the development of new antimicrobials.
Mm-hmm.
Because what this particular paper does is in describing the TAM system, or the translocation and assembly module, these folks are actually taking virulence determinants with them across the membrane.
Mm-hmm.
And they actually do that. We're sort of going out of sequence, but I'll give you the punchline is take a look at specifically figure 3, where they're looking at a set of mutants, the TAM-A domain, the TAM-A component and the TAM-B component in a Citrobacter rodentum experiment, in which they are specifically looking at virulence of this particular Citrobacter in mice. And Citrobacter rodentum in mice results in diarrheal-based disease process. So back to their story, I'm always amazed at how well Elio summarizes the paper.
Got a lot of experience.
He's got a lot of experience, and I think he's got his, Small Things Considered article sitting next to him.
Yeah, I think he tends to pick a lot of those. Yeah, it's okay. You could start a blog and do the same thing, Michael.
Yeah, and then I'd go, my lab would be closed because I'd have, you wouldn't have any grants then, and it would, sort of take over your life. So anyways, back to TAM. It's part of this OMP85, and OMP is outer membrane protein, and TAM is in the outer membrane, and TAM-B is in the inner membrane. And together, again, we got to do a little digression here about genetic nomenclature. When you see a lowercase letter in TAM, TAM with a big A, that's the gene. And when you see a big T, lowercase am in a big A, that's the protein. And the two of these proteins, TAM-A and TAM-B, get together to form the functional autotransporter, or the translocation assembly module, that then promotes the efficient secretion of autotransporters in Proteobacter.
Michael, so the title of this is Discovery of a Transport System, but it's my impression that these proteins were known already, right? Obviously, the genome is sequenced. Yeah. They know the protein. So what they've done here is to figure out exactly what they're doing. Is that what the discovery part is?
That's what the discovery part is. And, the way they decided that this was novel is they went after, or they did data mining.
Mm-hmm.
And what they effectively did is they did cluster analysis of sequence, or clans, in order to effectively put the TAM-A, TAM-B genes, which live in the same operon, which is always very cool to me, where, you have these genes. And again, one of the discussion elements that the authors bring up in some of their writings on their website and allude to in the discussion of the paper is that, this is found in many virulent gram negatives.
Mm.
And so what they ended up doing is they took Citrobacter rodentum as their model system, because it causes this wonderful diarrhea in mice. And then, like all good geneticists, they made deletion mutants of TAM-A or TAM-B. In a sense, it was in the same operon, and it was in a well-characterized gram-negative system. It was relatively straightforward to be able to make mutants in order to first do the subcellular localization to find out where these things go, And
so that's how they found out that one is in the outer and one is in the inner membrane. They did
correct.
Very nice. I was impressed to see some very nice biochemistry here.
Oh, this is a primer on the graduate student of the future. You have to be a card-carrying geneticist in terms of being able to analyze sequences. So you basically get your idea from doing the clans analysis, and then you have to be adept at the molecular biology to make the mutants in a model system that has a animal model so you can actually test your hypothesis. And I want to call your attention to figure 1C, where they have the mutant and the wild type, and they have what's termed a competitive index, where if you get a value, a competitive index value of 1, it implies that you have virulence. And, even, you don't have to be a very sophisticated graduate student to appreciate diarrhea, and that's a virulence of 1. And you look at their mutants, and the mutants in figure 1C all have virulence determinants or virulence competitive indices less than about 0.5. There's one mutant that one combination is just a shy bit above 0.5, but it's really neat and how they were able to address this aspect of deciphering the importance of this and being able to carry out the activity. And this is where they're looking at the beta barrel, looking for the bacterial surface antigen. And the sophisticated biochemistry that you alluded to was a technique that we may want to put the reference up or point them in the right direction, which was called protein shaving, which
protease shaving.
Well, they I think they called it protein shaving, but you effectively describe the technique because what you use is proteases in order to shave the protein and find out what's there. And again, they have magic machines in the sense that you have your mass spec at the ready in order to figure out what's actually going on.
So I like the way they did fractionation and found TAM A is in the outer membrane, TAM B in the inner, using anisir that they raised against the proteins. So it's really a lot of work here to sort this out.
There's an incredible amount of work in the 3 figures.
They do co-immunoprecipitations to show that these are interacting with each other.
I had a strong sense of deja vu because I did co-immunoprecipitations. Unfortunately, I didn't have available a mass spec back when I did it in the middle 80s. Because mass specs then were the size of chemistry departments and only available in chemistry departments. Today
everybody's got them. Yep,
everybody's got one on their desktop, and so it was really pretty neat and how they do it. And the meat of the manuscript gets into where they begin to demonstrate the importance of this subcellular localization is in figures 3B and figures 3C, in which they have 180 kilodalton protein, which is P1121, which in the GenBank, it was annotated coincidentally as an autotransporter. And here's where the novel discovery comes in, because they effectively did indeed prove that it was an autotransporter. And they then go on to talk about these autotransporters being adhesins. And
so if you mutate TAM A, they don't produce this P1121, right?
Correct.
So does that mean that this TAM complex is involved in the shuttling of P1121?
That's the sense that I got reading the paper, because the C. rodentum mutants lacking TAM A specifically did not express the 1121.
Right.
And so that's...
Of course, we don't know what 1121 does, right? We just know that it seems to be transported by this new membrane complex, right?
Transported by the new membrane construct.
Yeah, the TAM.
And then they used an assay in which they look for expression of adhesions AG43 and EHA-A, which causes aggregation of E. coli and promotes biofilm formation. And, the biofilm story is extremely important for our next paper because, the whole human microbiome issue. And as you begin to think about the why aspect of this, why do we really need to understand autotransporters in terms of, really advancing our understanding of being able to control microbes and being able to think about what we're going to be doing with the microbiome. If we really can begin to understand how to tickle the bacteria in the right way with autotransporters like TAM-A and TAM-B, making mutants so that you would either promote biofilm formation or depromote biofilm formation by sneaking a mutant into your probiotic that you may wish to be using.
This was such a cool idea. I don't know if it came out of the blue or if it makes sense, but they pick these two proteins that cause aggregation, and in a TAM mutant, the cells don't aggregate. So apparently these two proteins require TAM to get outside of the cell and make a biofilm. Am I getting that right?
You got that right.
Did they just land on this, or does this make sense that they would look at these two particular proteins?
I think it goes back, that's what you don't get. That's what you get out of going to a seminar where
yes.
where people can provide you with that perspective.
Yeah.
Today with manuscripts, running into the 3,000 word limit, you don't know why they did certain things. And my gut feeling is reading some of their writings on their website and some of their press releases from Monash University. These are our colleagues from Australia. Is they argue that TAM was discovered in many disease-causing bacteria, from microbes that cause whooping cough to meningitis to hospital-acquired bacteria that are developing resistance to many of the antibiotics that we are currently using. And so they're arguing, I think they went hunting for new targets, if you will, to begin to develop new antimicrobials in order to figure out how we can develop new antibiotics that may not be nuclear weapons in terms of our normal flora, but may just go after these transient members of our flora that are causing disease.
So your idea was to colonize with a mutant that can't excrete these adhesins, right? And
yeah.
That would prevent biofilm formation and pathogenesis at some level, right?
Hopefully. Hopefully. Or, be able to, tell the biofilm it's time to move on. This isn't a good place to settle.
Is that something we could do, Joe, you think, someday?
I think that's one of the ideas behind probiotics, is to develop competitive bacteria that can displace the pathogens or prevent the pathogens from developing a biofilm, as Michael says. I don't think anything's out of the question in the future, but the challenge we have is sort of related to what Michael was talking about in this figure 1, where they talk about the competitiveness index. And I think that's the crux of what we don't know, is what makes organisms competitive in a community. It's one thing to be competitive in a population within a species, but I think it's tough to study the invasion of a community, and that's what a probiotic has to do. It has to be able to penetrate an established community in the gut.
And I think that's why I think this paper is really neat, in the sense that it really describes this whole issue of beginning to develop a questioning system of how do you assess competitiveness. They were lucky in the sense that they had diarrhea as their readout because it's an easy phenotype to score. I think that goes back to all the, how, one of the, basic tenets of how do you do science is you have to ask the right question. And the competitive index type of question, I think, is going to become paramount as we begin to try to tinker to decipher this complex systems-based biology where we're no longer dealing with pure cultures, but we have a mixed community in which we're doing many things at the same time, and we're looking for an effect. And I think the secretion system is an interesting way because we know that in the case of something like E. coli, approximately 40% of their proteins that they manufacture in the cytoplasm go someplace else, whether it be the membrane, the periplasm, the outer membrane, or even out into the environment. And so they develop a substantial portion of their energy budget to moving things out to the outside. And 40% is probably a conservative estimate of how much stuff goes out, because we really don't have because we're dealing pure culture biology again, we really don't know what's going on in a community because we've been studying populations as opposed to individuals.
So, Joe, you work on a simpler system. Does that help you get at these competitive issues easier?
Yeah, it does, because we can screen mutants directly for the competitiveness or invasion phenotype, and we can separate those two. So, organisms may be able to compete with members of their own species, but they may not be able to penetrate a community. So by studying an organism that can penetrate, we can look for the negative. It's kind of interesting, though, historically, because when I wrote my first grant proposal when I was an assistant professor in the 1980s, which I hate to admit, I proposed to study the genetic basis of competitiveness in rhizobium, which is a very nice system because, as Michael said, the readout here in the paper we were just talking about is so clear. Rhizobium is a bacterium that infects plant roots and forms nodules in which it fixes nitrogen. So the nodules are like little tumors on the roots. And in many species of plants, only one strain of bacterium can get in. In some strains, lines of plants, more than one can get in. But no other bacteria, just rhizobia, get inside the nodules. And so you have this perfectly pure culture where, although they have to compete in the soil community with all the other organisms, the organism that wins out and ends up inside the nodule is in pure culture. So it's very easy to study. And my first grant proposal I wrote on this to study competitiveness, I got reviews that said, there is no genetic basis for competitiveness.
Great.
And my reaction was, what else is there besides genetics? And now we have Michael Schmidt sitting here on TWiM saying that we need to study competitiveness. So very satisfying. Thank you, Michael.
Happens.
You're most welcome. And I too have had grant proposals get reviewed in which I was ahead of my time. And it's unfortunate that that's one of the interesting things about science today is it's incremental and no longer taking the risk on the provocative ideas.
The grant proposal system has the problem that you can't predict often where the most interesting and important pathways are going to go. It's just very difficult. But the nature of it is you sit in a room and you decide where things are going, and you just don't know. So I think you have to put more faith in investigators than in systems sometimes, but that's not done, as you know.
Well, it's an interesting process that we have, and it's the best that we got.
So we have. That's right. They make a mention in the discussion of an interesting observation.
They're going to eukaryotes. Yes.
Well, that too. But before that, I saw they did a screen for proteins important in the pathogenesis of Klebsiella and Proteus, and they found mutants in TAM-B. All right, so you do an independent screen for what genes are important for causing pathogenesis. I don't know what the assays were, but you turn up mutations in this gene. So it's important, clearly.
And, Proteus mirabilis is this swarming microbe when you look at it on a Petri plate. And you can just imagine Proteus in something as complex as a biofilm. And Proteus is an interesting microbe because it causes urinary tract infections. And if you think about a UTI, where the microbe is literally holding on to the rocks on the Niagara River as the falls are coming down, it really illustrates in how this microbe has to move and hold in this. Well, unlike Niagara Falls, which is always flowing, the urinary tract doesn't always have liquid in it. So, it's able to move and attach and get itself in there. And similarly, if you think about Klebsiella, one of the hallmarks of Klebsiella pneumoniae is the amount of extracellular material it produces, more affectionately referred to as capsular polysaccharides. And, we refer to it as when you think it's snot, but it's not. And some of these extracellular polysaccharides are extremely slippery, and one of the ways the body deals with infections is phagocytosis, and the slipperiness of these extracellular polysaccharides effectively inhibit phagocytosis. So the microbe literally just slips out of the grasp of the phagocyte that is effectively trying to remove the offending agent from a particular area. And one of the reasons UTIs are recognized is because of the invasion of pus, which is phagocytes coming in. And so I thought the TAM-B component, we had TAM-A for the Citrobacter story, and now we have TAM-B. And again, there's only so many ways to make proteins that have structural activity. We know about structure and the importance of structure from our understanding of 16S ribosomal RNA, because that has to assume a structure in order to have activity, and we know where mutants are, and that's the basis of 16S taxonomy. And so these beta barrels that are being formed and the amino terminal ends that facilitate this may have the ability to really influence pathogenesis. So you can immediately see why this group from Monash University is so interested in using this observation to disarm disease-causing bacteria, and they're probably trying to design inhibitors that will go after disrupting the beta barrel, or going after sucking the N-terminal N into an alternative topology or something along those lines. And
you're plugging up that nice door that you call it?
Yeah, you're effectively eliminating it. And one of the things you really need to appreciate is what this effectively is doing is putting a screen door on a submarine, because any of you who have ever thought about submarines recognize that they have to be able to withstand tremendous pressures. And the pressure of a bacterial cell is at about 80 pounds per square inch from the inside to the outside. And so the bacterium is literally trying to explode all the time. And if you have this opening, you can immediately imagine how, if you just open the door, you can really easily kill the microbe.
Michael, I might use that for the episode title, Screen Door on a Submarine.
Yeah.
I like that. The last thing we should touch on is this interesting statement they make that maybe an ancestral TAM was the bacterial starting point from which the mitochondrial import pathway was established. This is based on some homology, right, Michael?
Again, this is the primer for the modern graduate student. You really have to be adept at looking at sequences and appreciating how you can compare and contrast genomes. And I thought it was rather interesting. They talk about this in the plastids as well, and the outer membrane of the primitive protozoan Trypanosoma brucei. And here the TAM protein is operating in the reverse, because remember, mitochondria are sort of the wrong way round. Because they're inside our cells, they're effectively bringing things into the mitochondria rather than moving things out of the mitochondria.
Right. All right. Anything else, Michael, before we move on?
No, I just commend the authors for their interesting work, and I commend it to the listeners of the program to take a look at it. It's an interesting read, and it's provocative enough if you begin to think a little bit outside of the box.
All right, thank you.
Michael, is there any evidence that there are homologs in gram-positive bacteria? I couldn't find any when I did a search, which I guess would make sense because of the membrane structural differences, but I was wondering if you knew of any.
I don't know of any off the top of my head. I went through, I did a quick and dirty search looking, just like you did, Joe, looking for the gram-positives, but I'll bet that there's probably something similar that the gram-positives have to getting things out. Again, I always go back to the whole issue of natural competence and how something as big as DNA is able to get inside a cell, and what the gram-positive bacteria do to become competent. And these are things like strep pneumo, is they make peptides that effectively make, install the screen door in their membrane and their very thick peptidoglycan wall that facilitates DNA entry into the cell. But they sort of cheat in the sense that, remember, in transformation with competence, always one strand of DNA is burned as the energy. So you sort of have to pay the bouncer at the door in order to get the DNA in. So you have to grease the palm in order to get it in. In biology, there's no free lunch across the hydrogen-tight membrane.
Oh, Michael, you're on a roll today. It must be being back in your own office helps, right?
And I'm not having anxiety attacks over the Wi-Fi connection, or will Ray go crazy trying to put us together?
Our next story has to do with the gut microbiome, and this I noticed in Nature in a news article this week. The title of that was "Gut Microbial Enterotypes Become Less Clear-Cut," and there's some results from a meeting that we should talk about. But I thought we should actually go to the original Nature paper published last year called "Enterotypes of the Human Gut Microbiome," which we never really have discussed. And maybe Joe could take us through that.
Oh, I thought we did discuss it.
Did we?
I think so.
I think we did.
Yeah. So I wasn't going to do it in enormous detail because I think we did discuss it, but I will.
Say we have. Maybe I wasn't on it.
You may have been on vacation.
Oh, yeah, I think that's right.
You were on vacation, Vincent.
Could be. I'm going to check it while you just briefly summarize that for us then.
All right. Well, if you missed it, then other people did too, so we should spend more time on it. So about a year ago, there was a lovely paper in Nature that showed that bacterial communities from the human gut appeared to come in 3 flavors. And this was a study of people from all over the world. They sampled on 3 continents, and there were 39 people in the study. So that's one of the issues that we have to be cautious about. It's only 39 people, and there's probably a lot more variation among humans than can be represented among 39. But despite that, they found that among the 39, there were 3 enterotypes. And an enterotype, in their definition, was a cluster of species or a cluster of genera that occurred together repeatedly and were the dominant organisms in a particular enterotype. So an enterotype of one type, for example, was dominated by the bacteria Bacteroidetes or Bacteroides. And in a second one, they were dominated by Prevotella. And what was striking is that they showed that these same 3 dominant groups showed up over and over across every variable they could look at, geographically, diet-wise, sex, anything they could associate with genes. They could find no real patterns of what would dictate these 3 types. but they kept finding them on the 3 continents, on all 3 continents. So that suggested that there was something else driving the formation of these clusters of types of organisms that showed up in some humans and not others.
So in this 39-person microbiome study, were they all healthy individuals?
Not entirely. They correlated the one member of the cohort who had inflammatory bowel disease, and then there seemed to be a representation of obesity, but I couldn't tell from the paper how many people actually were obese. And they just plotted those in the rest of the communities. So it didn't seem like they stood out as highly distinct. But again, there weren't enough samples to really say.
Of course, many people are interested in how certain conditions affect the microbiome, and those studies are going on all over the place now, right?
Yeah, there are many of those, and I think we're going to be getting some fabulous data from the Human Microbiome Project on exactly that.
Great. And did we know at the time or since then when these enterotypes are established in life? Is it very early on?
That would be the guess based on other work, that the enterotypes begin to form probably when the community that looks most like the adult community forms. And that's pretty early in life. Infant communities change very quickly as their diet changes and as they get older. And then the community is more stable. It changes, certainly with diet and with other factors, but it doesn't change as much between childhood and adulthood.
And I guess you're influenced by your parents as to what kind of enterotype you would get?
Well, we know that the gut community is influenced by the mother's flora.
Mm-hmm.
We know that to some degree it's influenced by the people that one lives with. So that would not be genetics, that would be environment.
Mm-hmm.
It's definitely influenced by diet. Some work has shown that diet can radically change the community within a week. And so the difference between, for example, what one of our colleagues calls this the Western diet, which is represented by McDonald's. Maybe I'm not supposed to say names on the air, sorry. But the classic Western diet that is high in fat.
Fast food, right?
Yeah, fast food diet that's high in fat and sugar and salt has a radically different composition from the classic hunter-gatherer diet, or a particularly high fiber, high carbohydrate, but low fat and low protein diet. So there seem to be multiple influences on the community, and obviously some of them are genetic, but it's not the whole story. There are definitely environmental influences.
I think we've done some more recent paper where they looked at the effect of changing the diet from what you call a Western to a hunter-gatherer diet. And then, if I remember, correct me if I'm wrong, there are rapid changes in the microbiome when you do that. But I don't remember if you change the enterotype completely. Do either of you recall?
Well, actually, a lot of the studies on diet were done before these enterotypes were defined.
Okay.
So I'm not sure. I haven't looked at statistical analyses that analyze the older data by the enterotype types. So I don't know that. It's a good question.
I think what's going to probably need to be done is sort of a Cochrane-type analysis of some of these papers to begin to try, and I think that's what the multi-center folks are doing for the NIH microbiome project or the worldwide microbiome project. I think they're going to probably begin to, hold conferences where they begin to decide how they have to reduce some of these data into really asking the question you just asked, Vincent, to really try to begin to make some sense out of it, especially when you consider the paper that we discussed on TWiM number 30, which was looking at TLR2 knockouts, albeit in mice, and the whole issue of the microbiome type in that particular genetic lesion and the whole incidence of type 2 diabetes. And,
right.
You begin to look at this typing system, and then you begin to consider something like the genetic background of the host and there are genetic components in the host that predispose people to type 2 diabetes. And so you almost really want to begin to say, well, we really should be developing diagnostics that can first tell us our enterotype. And I think when we discussed this paper the first time, is there were folks sending their microbiome to folks, or there was a company trying to be able to tell you what enterotype you are, so you could try to influence a better lifestyle so that you could improve your health.
Right. So the paper I was thinking about, I just found it. We talked about it on TWiM 19. It's called Linking Long-Term Dietary Patterns with Gut Microbial Enterotypes. It was published in October 2011. And they did a controlled feeding study of 10 people only. And it showed that the microbiome composition changed detectably within 24 hours of doing either a high-fat, low-fiber, or low-fat, high-fiber diet. But the enterotype identity remained stable during the 10-day study.
And they used the same enterotype definition as in the paper we were just discussing?
Right.
Okay.
Bacteroides and Prevotella, just the two, I guess. In that paper that we're talking about now, there are three, right? There's a third.
Right.
And I didn't look at that. But so the answer is, it's within a short term anyway, it's stable. So I guess you need to do it. If you did it for a long term, maybe you would switch the enterotype.
Mm-hmm.
Anyway, all of this may be moot because, according to this meeting, may not be so clear-cut, right?
So, yeah, it's kind of interesting because I think the report on the meeting makes it sound like the results are more opposing than they actually are. So just to finish on the original paper, they did a metagenomic analysis, which means sequencing the full microbial complement of the community, all the genomes together, and then looked for dominant organisms as well as dominant genes and defined these enterotypes. And I think when we talked about it at the time, we were all kind of amazed because there was no correlation with any of the parameters that you might imagine. Mm-hmm. They called them the cross-national clusters because there was no obvious geographic delineation. Another thing in that paper that was very interesting was they tried to develop correlations between body mass index, or BMI, and the type of community. And surprisingly, they didn't find a correlation with the enterotypes. And that was inconsistent with previous work that's shown that the ratio of the firmicutes to the bacteroidetes, which are two major phyla represented in the gut, seems to be associated very tightly with obesity. So high firmicutes and low bacteroidetes seem to be associated with obesity. The opposite seems to be associated with thinness. And there are a number of other correlations that associate with the ratio of those two groups. This study didn't seem to find any correlation with obesity or BMI and the types. So it was a really interesting study and very provocative at the time because it was inconsistent with, or it certainly raised questions about some previous paradigms, but also laid down these three types as kind of a very strong structure as they presented it for thinking about the types of communities that humans can have.
Well, Mike, one of the things that I reacted to was, why would there just be three? Why that number? Why not 5 or 10 or 1 even, right?
Yeah, I mean that was, I think, what we all were stunned by, that okay, it was only 39 people, but across this incredibly diverse population to find only 3. But one of the reasons might be that they were defining the types at a very high level, and using very abundant organisms. And so it may be that when you see these shifts from one type to the other, you're looking at a pretty gross level shift. And perhaps if you look at a finer level, there'd be 5 or 10 or perhaps 1,000 different enterotypes.
I see. Okay. So it's all in the analysis, really, or it could be in the analysis that it makes increases the number. Yeah. I think they made a mention at some point that the minority populations contribute a great deal to the types. If you look at individual genes, for example, like they were talking about pili. Yeah.
Yeah, that was one of the interesting contrasts, is that although they seem to focus on the dominant organisms or most abundant organisms in defining the types, the presence of genes, particular genes within the enterotypes, was much more complex than just three. And so, yeah, they mentioned that, for example, E. coli, which is a very low abundance member of the gut community. In fact, it's almost rare in the gut community based on total community membership, contributed something like 90% of the pilus genes to the community. And what that means, we don't know, except that it's clear that very low abundance members can have a big influence or can be contributors to one particular element or feature of a community.
Mm-hmm. Right.
So the recent report was a news report also in Nature that came from a presentation at the International Human Microbiome Congress in Paris that occurred recently. And in that report, which we don't have the data for that because the paper's still in press, so we're basing the discussion on much more general reports of it. What they found was that the ruminococcus piece of the story, the original story, which was one of the three microbiomes or enterotypes, was now complemented by another archaea called Methanobrevibacter. And so by looking at a larger population, all of a sudden this new organism showed up as one of the dominant types. So that in itself, I thought, was pretty intriguing, that this particular group, this genus of archaea, was not discovered, at least that I saw in the original paper. It wasn't mentioned. So that already makes you wonder, is it the number of people in the cohort or where they drew their sample from that there's such a difference? This study used 663 people as opposed to 39. They were all from Denmark and Spain. So it was a slightly different but somewhat overlapping demographic, but again, a much larger group. They also did total metagenomic sequencing, and they made it sound in the report like the lines were getting very fuzzy between the enterotypes, and that maybe there was no such thing as these three enterotypes. But if you look at the data or the discussion of the data, the current study does seem to back up that there are two enterotypes that are clearly different, one dominated by Bacteroides and the other one dominated by Prevotella. And those are kind of at two extremes. And then it's in the middle where the third enterotype seems to lie, that is, I think, a little bit more complicated and may have fuzzier lines. And so that's this ruminococcus Methanobrevibacter.
Mm-hmm.
And remember that about a third of us, at least in the United States, are methane producers. And the archaea are the only group that make methane. And so you'd have to have the Methanobacter in you in order to be one of these methane producers. And so you begin to see if you have a cohort of 39 and about a third of the population does indeed make methane, and you can do this experiment in your lab if you have a GC, you just blow into a tube and look for the evolution of methane out of your breath. You can detect whether or not you do indeed have this methanobacteria. But the question that I had about this methanobacterium or methanobrevum is whether or not the extremes like Prevotella and the Bacteroides actually had methanobacteria in it, and whether or not that's distributed across the three enterotypes, or it was just defined to the ruminococcus. And, not having had an opportunity to go through all the sequence data, and again, it may have been underrepresented in the population, so it just may not have made the hit parade when they were doing their metagenomic sequencing.
In the first paper, they do show presence of Methanobrevibacter in actually the community that was dominated by the Bacteroides. And so they did find it, but it was not a main contributor, as they called it. So
Right.
it was a co-occurring contributor. And so that was kind of interesting. They didn't seem to see it as synonymous with the Ruminococcus, which is what the current group found.
They also have a quote from another person, Dan Knights, who had looked at 1,200 adults, right? I guess he reported at the same meeting.
Yeah, and I think that work is also in press,
Yes,
and
right.
he has a much larger sample. The original paper also said that there was a report on the original paper that said that the authors of the paper from last year had another study coming out with 400 people. And I didn't have a chance to look for that, but I wondered if they were able to corroborate the original 3 enterotypes with that data set.
That's not out yet, though, right? Is that right?
It didn't seem to be.
Yeah.
I didn't find it, and I didn't have a chance to write to them to see if it had come out.
Yeah.
So I think there are going to be a couple more really big studies that may address some of these ambiguities or places where there might be overlap in the enterotypes.
We'll have to talk about them when they come out, I think, right?
I agree, definitely.
This is just a little tantalizing information. And we're basing it on how the writer, of course, has interpreted the presentations as well, and it could be different in a published paper.
Yes,
For sure.
absolutely.
What you say at a meeting is often different from what you eventually publish.
Right.
It's those pesky reviewers.
Yes, well, that would be us, right? But it's not surprising that as you look at more numbers, you find the curve smoothing out, right? Because 39 is a very small number of all the people in the world.
And unlike mice, people are not inbred. And so you would expect differences where, 39 mice is, a lot of mice, whereas 39 people aren't a lot of people.
I don't know. In the population of world mice, maybe 39.
Well, no, I mean,
I know what you mean in the lab.
It's because of the genome of the poor little mouse.
Sure, got it. Just kidding. We should also remind everyone that Joe works on gut microbiomes in caterpillars, right?
Yeah, much simpler than these horribly complex mammals.
I think you're about to publish another paper, and when you do that, we'll talk about your work because I think we can learn from it. In simpler systems, often allow you to make different conclusions more readily.
Okay, we can talk about probiotics sometime.
That would be great. We do actually. Anything else, Joe? Before we move on, is that basically it?
Yeah, that's about it.
We do have a letter from Peter, which is relevant to this discussion, actually. He writes, Dear TWiM team, a fascinating article from New Scientist this week. Standard medical teaching is that the fetus is sterile and that the microbiome only begins to develop postnatal. New research from Spain indicates that the microbiome starts to develop before birth. So this is an article in the New Scientist, and I don't have access to it, so I couldn't read it, but he sent a summary, and it says, Pilar Francino and her colleagues at the University of Valencia in Spain collected and froze the meconium of babies from 20 women. They removed the outer layers of each sample to rule out any bacteria picked up after birth, then looked for bacterial DNA. The team not only identified bacteria in the baby's meconium, which before then was thought to be sterile, they found bacterial communities so developed that they seemed to fall into 2 categories. Around half of the samples appeared to be dominated by bacteria that produce lactic acid, such as lactobacillus, while the other half mostly contained a family of so-called enteric bacteria, such as E. coli. So that's interesting, but of course we don't have the paper to look at either. So does that surprise you?
It doesn't surprise me. I've been talking to my friends in neonatology, and the neonatologists have been keeping this under the radar amongst themselves, but to them, this is old news. They've been talking about the last trimester and bacteria moving into the infant or the fetus that will then become the infant. And I guess intuitively it makes sense because if you think about it, at day one, you're able to digest an extremely complex food called human milk, which is, really complicated.
So these bacteria would be entering during fetal development. Is that the idea?
That is the idea.
How would it get there? I mean, the blood is not bringing it there, right?
Yeah, I've asked that question, and they shrug their shoulders, and they say, well, that's what everyone is trying to understand. Whether the placenta becomes semi-selective, where certain bacteria can move across, and, thinking about transport, if bacteria can transport proteins, can the placenta begin to transport bacteria? Because if you go back and you look at some of Brett Finley's work about how salmonella locally invades and gets inside the system, you can begin to think how macrophages can begin to move bacteria. But again, the whole blood barrier is rather interesting. So I think that's going to be an interesting story to follow.
What do you think of that, Joe?
I think that sounds like a really interesting possibility. It's a little inconsistent with all the work on gnotobiotic mice. So these are germ-free mice where, as I understand it, the colonies are developed by delivering babies by cesarean section, and they come out pretty sterile. Not pretty sterile, they come out sterile apparently.
They come out sterile.
Yeah, I mean, otherwise the ntobiotic world would not work. And so it makes me wonder, is that a human mouse difference or something else? Because that does seem to be a pretty strong trend in other mammals, at least mice. And then with humans, we certainly see a dramatic difference in the microflora that develop in the guts of cesarean section versus vaginally born babies. And that's thought to be due to the bacteria they pick up from the vagina. So again, it makes me wonder, how much are they actually exposed to in utero, and how much, is inoculation as they're born or after they're born?
Well, we'll keep an eye out for the paper.
But the gnotobiotic animals come from a sterile mother too, don't they?
Well, they had to start someplace. So I think they try to reduce the populations in the mother as much as possible. But my understanding was that if you deliver by cesarean section, even in a mother that has bacteria, the infant would have no bacteria. But maybe that's not right. So we'll have to find that out when we look at the paper.
Yeah, we could pester people like Ed Baelish, the gnotobiotic guru, and ask him for an opinion.
Yeah, that would be great.
Well, thanks for that, Peter. That's interesting. We have another email from Peter, who may or may not be the same one. I don't recall the email address. It must be, he writes, Dear TWiM team. Yeah, it's got to be the same Peter. Interesting article in March Scientific American on long-term effect on health that food poisoning can cause. This is a Scientific American article called Food Poisoning's Hidden Legacy. It's by Maren McKenna. Basically, the idea here is that studies in several countries show that food poisoning, rather than lasting just a few unpleasant days, can in some cases cause lifelong problems. Though this possibility has been known for some time, the recent work indicates that the phenomenon is much more common than previously thought. This is a survey of 100,000 plus residents of Sweden who had food poisoning between 1997 and 24, found that they had higher than normal rates of aortic aneurysms, ulcerative colitis, and reactive arthritis. And another outbreak in Spain of salmonella, similar ideas. What do you think about that?
That's a scary possibility if our guts are being changed, or our health is being changed for life by food poisoning outbreaks, given the frequency with which most people get food poisoning in modern food preparation. That's a little frightening.
It is.
Considering the average, there are I think the number is 77 million Americans get food poisoning each year. So that's almost a third of the country.
Wow.
I would think it would be hard to tease out whether food poisoning or something else caused your illness, right? With that number.
Yeah. And because food poisoning is so common, how do you know it's the same and not you're just being reintroduced to the same superantigen? Because the common ones like staphylococcal food poisoning is actually an intoxication. You actually eat the toxin, and it is a superantigen, and it can manifest itself. And as we know from our good friend Staphylococcus, its superantigens do many remarkable pathogenic processes in our, from reactive arthritis to muscle pain, stiffness, because superantigens induce the lovely cytokine response. And, a lot of these symptoms could just simply be, a reintroduction. And enterotoxins are really pretty common.
There's a wide range of illnesses associated with them, heart problems, colitis, arthritis. So, it's hard to understand mechanism based on that. We'll look for the paper when it comes out, see what happens. Peter says, I suggest you have Maren McKenna as a guest on TWiM or TWiV. In fact, Maren was at ASMGM last year. She sat through the, or was it ICAC? Yeah, it was ICAC. She sat through TWiV at ICAC. She said it was interesting. Either of you know her?
I'm going to talk with her tomorrow.
Are you getting interviewed?
Yeah, I'm getting interviewed.
On Copper.
On Copper.
Yeah. I think she's a pretty good writer. I read her in Wired now and then. Do you know her, Joe?
No, I don't. Yeah, I have to look her up.
I think she does. She wrote the book, what is it, Superbug? Is that it?
Oh, yeah, of course.
Yeah. Our next one's from Nathan, who writes, thanks for the great podcast. TWiM is one of my new favorites. I'm about to post a review on my blog. I'm a little afraid to start listening to TWiP and TWiV. If I get hooked, I'll never be able to keep up with all of them. I just listened to your episode that included the paper on the autism microbiome study, and one of you said something about it being difficult to recruit controls for this kind of invasive procedure. It might make the statistics more complicated, but what about recruiting siblings of autistic kids as controls? The parents would be much more likely to be on board, as they have a vested interest in the progression of science on the problem of autism. This was the paper from the Lipkin lab here, where they found a new type of bacteria in the guts of autistic children. You remember that one, Michael?
I do. That was the Sutterella paper.
Sutterella, right.
Which is formerly Bacteroides before the taxonomists renamed it.
And we were mentioning how, so these are based on biopsies done through colonoscopy. So not easy to get volunteers for that kind of study.
They're going both ways, from the top and the bottom.
Yeah. So what, does this make sense to do a sibling? I don't really know.
The whole issue of informed consent and, it's really one of these ethical quandaries where, because there's anesthesia involved in this, and so it's really a challenge, because anytime you undergo anesthetic, you do incur a risk. And the principal risk, which is 1 in 1,000, is perforation of your colon.
Right.
And so that is a significant risk. Having just had a colonoscopy recently where they explained to me the risk, I was surprised at how frequent perforation of the colon was.
Yeah. You shouldn't say it, because people need to get them, but you don't want to scare them.
They need to get them. I got one. It was not bad at all. The preparation the evening before was
it's unfortunate, yes.
Was relatively vigorous.
It is. The prep is the worst part, because you're asleep during the procedure. But yeah, I know someone who was perforated. She woke up and they said, we have to bring you into surgery right away. Actually, she may not have awakened. They might have brought her right in to fix it. So it happens, but...
Yeah, it seems that doing it on children without a cause except for research would not be something that most review boards would approve, or parents for that matter. I can't imagine the parents approving it.
And then, of course, if you only look at kids who are having it for some other reason, you're biasing the study, right?
Sure.
Yeah. So it's a challenge, but that's the whole issue with informed consent. You have to consider all angles.
Yeah. All right, our last one is from Steve, who writes, "Hello, Vincent and cast of TWiM. I have to say, even though I am a novice, I really enjoy your podcasts. Because of the nerdiness and nature of the song, I thought I should pass it along. Happy St. Patrick's Day." So he sends us a YouTube link to a fellow playing a song all about beer and Saccharomyces. And it's fun, so check it out. St. Patty's Day was a while ago, of course, but it's still relevant. And that will do it for email. Of course, we love to get your email questions and comments. You can send them to TWiM, TWiM, at twiv.tv. Of course, you can find TWiM at iTunes, the Zune Marketplace, and also at microbeworld.org/TWiM, where we keep all of our show notes. If you like TWiM, go over to iTunes and you can rate it. You can give it a couple of stars. Give it 5 stars if you like it. And that helps us stay more visible on the front page of the medicine podcast directory over at Apple there. I'd like to thank everyone for joining today. Joe Handelsman is at Yale University. Thank you, Joe, for coming today.
Thank you, Vincent and Michael. This was a really interesting one. I learned a lot.
We do miss you when you're not here. I wish you could come every time, but whenever you can, we appreciate it.
All right, I'll come more often with an invitation like that. How can I resist?
Anytime. Elio always says, "Is Joe going to be on today?"
Oh, he's so sweet.
He would like you to be on more often. Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
As always, I learned a lot.
Good to hear you in your office. It's so much better.
I agree.
Just don't travel.
Just don't travel.
Tell him I can't. It's because of TWiM.
It's either that or get a much better connection.
Someday you'll have a little card where you'll get a huge bandwidth connection and you won't have to worry about it.
That's true.
The hotel internet will no longer be popular. I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for supporting TWiM, Communications Director Barbara Hyde, and Chris Kondayan and Ray Ortega for their technical help. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology. Did you hear about there was a CDC report today, a plane leaving Madison or somewhere in Wisconsin, I forgot where. There was a bat on the plane.
Oh, no.
They discovered it shortly after takeoff, and they returned to the airport. This was in Morbidity and Mortality Weekly Report. And the bat hid in the bathroom. So they got everyone off the plane. They opened the door. The bat flew out of the aircraft. It flew into the airport and out the front door and escaped.
Now that's a smart bat.
And that bat's offspring are going to be really good. Isn't that great?
Wow.
Do they have to vaccinate everyone for rabies?
So they found out that no one had been bitten or scratched by the bat, so they decided they didn't have to do that.
Well, it's not so much that, it's the saliva and the ultrasonic generation. Because in South Carolina, what the public health people tell us is that if you sleep in a room with a bat, you need to be vaccinated.
What?
Mm-hmm.
If you sleep in a room with a bat, you need, at least in the South Carolina bat population is all rabid. And so consequently, you need to sleep in a room. If you sleep in a room with a bat, you need to be vaccinated for your own protection because there is no treatment for rabies.
Yeah, but I've never heard that. And actually in Madison, I heard many stories about bats in people's bedrooms and going after the bat with a tennis racket in the middle of the night. And that is amazing that they...
Well, I'll confirm this, but that's what our public health microbiologist tells us. So I'll double check with him.
Because I think they're all rabid in Wisconsin too, or at least very high rate.
So they didn't get any saliva or any droppings. They didn't have the bat, so they couldn't test anything.
Yeah.
They don't know the rabies status. But so as far as I can tell, they decided not to immunize everyone.
Yeah, because it's an expensive vaccine.
And I think 5 people left, and they don't even know
who they were.
I don't know how that could be in this era, but there you go. Anyway, I just thought of that when you said Wisconsin.
Oh, well, I'll have to look that up.
It's in this week's Morbidity and Mortality Weekly Report. It's
Since my husband is on a plane right now going to Madison. I have to worry about his plane. We'll hope that there are no more bats.
Actually, it was It was from Madison, bound for Atlanta, Georgia. Yeah.
Yeah, that's what you said. Wow.
Shortly after takeoff, a bat flew from the rear of the aircraft through the cabin several times. Can you imagine?
No.
Can you imagine the hysteria on that plane?
Especially if it's one of those small ones, which it probably was coming out of Madison, right?
Yeah,
yeah. It was probably a cigar plane. Yeah.
You get pretty close to that poor bat.
Yeah.
Wow. Well, I'm glad it emerged unharmed.
I'm
That's
actually
good to
glad
hear.
the bat got away. I kind of like bats.
Yeah.
All right. Thanks again, everyone.
All right,
All right.
Vincent.
Bye-bye.
Have a good
See
weekend.
y'all.
You
Okay.
too.
Bye-bye.